Hello everyone, my name is Bill Grabert. Welcome to the monthly Tuleyome Nature and You lecture series for August 2020. Tuleyome is a nonprofit conservation organization based in Yolo County, California. Our focus is the northern inner coast range of California. It's the area of the Berryessa Snow Mountain National Monument. These mountains just to the west of the Sacramento Valley are remote and generally unknown to many people. Our goal is to conserve, educate, and generally encourage people to enjoy this interesting area. Much of it is public land. You can learn more about us by going to tuleyome.org. Tonight's talk is about ringtails, one of the most charismatic animals in our state and generally a very difficult one to spot in the wild. The speaker is David Wyatt, a professor of biology at Sacramento City College, who teaches a variety of classes in the field ecology program. Dave just completed a nearly decade-long study of ringtails in the Sutter Buttes, a small, distinct mountainous area in the center of the Sacramento Valley. He's also very familiar with the ecosystems of Central America and is working on a cataloging project of insects and bat species in the mountains of Belize. I first met Dave in about 2015 on a field trip to the Sutter Buttes and got to see him in action with his students sedating, measuring, tagging, and releasing captured ringtails. His enthusiasm for his work is very contagious, and we are very honored to have him with us tonight. Welcome, Dave. And now I will turn it over to you. Thanks.
Excellent. And everyone that's actually on the talk, please make sure that you support Tuleyome. They're a great organization. They're having to deal with a lot of fire issues right now, of course, but it's a pretty spectacular wilderness area, and there's ringtails out there, so that should make you want to at least consider supporting them. So it's I feel honored that Bill asked me to give this talk, and heck, I'll talk about ringtails all day. Ringtails are not necessarily a charismatic species. Yeah, I'm lying there. They actually are, and there's something that most people don't know about, which is really remarkable. And I've had the tremendous fortune to be able to work with these guys. My first introduction was hearing about them in 1984 in an ecology class I was taking at California State University, Sacramento from Gene Trapp. Now, it turns out Gene has had the number of graduate students that have worked on ringtails, and by December of 1984, he made mention that a grad student named Mike Lacey had a couple of collars still out for ringtails, and that he was looking for a volunteer to go out and canoe out to a site and to live trap some ringtails. And I thought, what the heck's a ringtail? And I was working at Payless Drugstore at the time, and thought I could take a weekend off. I might as well do that. So I volunteered, and I gotta say it was an amazing experience, because the first ringtail we caught, the first one I ever saw, was missing one eye, was missing one paw. And it might have been like the snap traps or something that they were still using at that time. And it was the most beautiful animal I ever saw. I was stunned. I had no idea they were like this. And we're talking about something that's about 2/3 the size of a full-grown house cat, weighs about 1,000 grams. So we're talking about something around a half pound in that neighborhood. Yeah, is that correct? 2.2. Yeah. So around a half pound, and they are an incredible animal. That's a little mesocarnivore, and they're not only that, they're charismatic, and no one really knows about them because they're strictly nocturnal. So what I'm going to do is, I see that we're starting to record already, so that's cool. I'm going to actually share the screen with you, and we're going to talk about ringtails. I'm going to hold that screen open and tell you a little bit about my background, and then we're going to dive into this. And this is the culmination of about 10 years worth of research that's been going on in the Sutter Buttes. So let me do the share screen. We'll do the desktop, and I've got some videos in with this as well. So let me open this up into a full screen. Awesome. So now what we have as a title is a ringtail ecology in the Sutter Butte, Sutter County, California. So I teach at Sacramento City College in the field biology program that's in the biological sciences department. And I teach a variety of classes. Every semester I teach natural history, and I also teach environmental biology. But in the fall, I also teach field methods in ecology and also restoration ecology. And then the spring, I flip those two out for environmental regulations and introduction to entomology. I also do a number of field programs like the Baja Field Studies program we did for a number of years. Now it's based out of Glendale College. I don't teach that any longer, but we work with Glendale on that. And then also the Mojave Desert Field Studies program. And it's sort of sad, this May was the first time in 24 years I haven't done that. So I'm sort of missing the desert. Maybe not right now because it's 120 degrees. But still, you know, we get ringtails down there as well. So get to see that. I work with mammals. I graduated from California State University, Sacramento from my bachelor's degree and master's of science degree in conservation biology. And I was very fortunate, like I said, getting to know Gene Trapp, and the influence he's had on my life is second only to Bruce Swinehart, who basically converted me from a criminal justice major into a biology major, and when I took his natural history class. And the irony of it was the natural history class I took at American River College from him, and then the following semester I took environmental biology, ended up as the two classes I was hired to teach at Sacramento City College. And Tom Scandoni was teaching those, and he was the Bruce Swinehart over at Sac City. I worked for 10 years with the state of California as a biologist with California Department of Fish and Wildlife, Fish and Game, as well as Caltrans. And then I've maintained my own consulting company since 1996, where I specialize in bats and in ringtails. So that's a little bit about my background. I've lived in California for all but 7 years of my life. And during those 7 years, I was in the air. My dad was in the Air Force and traveled between, let's see, I was born in Hawaii, spent a few months in New York, a few months in Kansas, about 5 years in Japan. And then he got stationed here in North Highlands at McClellan Air Force Base. And then I've been here ever since. I do eco tours. I lead eco tours to Belize and to Costa Rica, Panama, as well as Ecuador. And we're adding in Brazil, hopefully. We'll see how COVID goes. So those are some things that I've been able to take a number of people to, and that's been fun. But, you know, one of my favorite places on the planet is this place right here, Sutter Buttes. I love this place because it's this large parcel of land that's this volcano which came up about 1.5 to 2 million years ago in the middle of the Central Valley and Sacramento Valley, and it creates a circular structure that is about 10 miles in diameter and is made up of private lands with the exception of a small parcel, about 1,000 acres of state land in the middle of it. But it's a lot of it's in conservation easement as well. And the ranchers have been fantastic in giving me access to the land. And in particular, this particular parcel is called the Dean Ranch. And we have the Middle Mountain Interpretive Hikes, which allows people to go in on a guided hike. You know, you go with a guide, you stay with the guide, and you get to have these wonderful hikes to even the top of North Butte, which is this picture here. And this picture is significant because a lot of the data I'm going to show you is from this location, because the results that we got from this location of 11 ringtails was just mind-blowing, you know, and that led to going from initially a 1-year study into a 10-year study. So from here, let's move on and talk about the taxonomy of ringtails. Ringtails are in the order Carnivora, so they share the same order with dogs and mountain lions and bears, as well as raccoons. And the Procyon family are the raccoons. And this is a ubiquitous guy that we see around even downtown Sacramento that make little territories. But we also have the kinkajou, which is also known as the honey bear. And we have the coati, which is also a very cute little critter right here. This is a baby coati. So they're fun. And I'm actually going to end my talk with a video of a baby ringtail. So if I don't have you hooked by then, that will hook you on ringtail charisma. All right, so here's some pictures. I'm going to start off with pictures, get you guys really hooked in before I show you some of the scientific details. But This is a guy named Jack on the left. He crawled right up into this manzanita and took a little rest and gave me a chance to take a picture like this, which I absolutely adore this picture. And one of my first ringtails is Star, which is on this upper right corner, and she made this little climb up this tree, and then you don't see her tail here other than this little end of the tail, but this is a beautiful picture showing that amazing face that they have with the reflective properties around the eye, those huge ears that can swivel and catch sounds, and then just look at these eyes, just are made to provide really good vision in nocturnal environments. With the vibrissae and the whiskers, it gives them multiple opportunities. And here's another ringtail. You can see the ear tags in this one. So I am doing tagging on these guys using these small Monel number ones. This is Mike, was a name I gave this one. This one happens to be Ben, and this is Sylvia. It's sort of easier to refer to them that way than 2008-11-21-A. Yeah, I might anthropomorphize them a little bit, but it helps with identifying who's who. This is Star again, and Star made it to, I have a cat that's rubbing against me now, but Star made it to the Peterson's Field Guide. So I was pretty flattered when Fiona Reed, who I had gone with down to Belize on a bat expedition, and that was amazing. We caught like 540 species of, or individuals of bats from like 28 species. And she was revising the Peterson's Field Guide. So she asked if she could use one of my photos, and this is the one that she chose. So it definitely shows that ringtail aspect of it, most definitely. And this is Katie on the left, and this is Katie coming back down the tree. So you can see the top of it. Top of the tail is much more darker than the underside. The underside has a nice white band. And what they'll do too is they'll throw that tail over their body as they're crossing over open areas. And they are excellent climbers because, like house cats, they have retractable claws that come out. And same way that gray foxes are really good climbers too. So these guys are definitely good climbers. They are dead silent when they're moving around. It is pretty phenomenal how quiet they are, and all of a sudden you see eyes shine and there's a ringtail. So here's some more views of some ringtails. This is the bottom one is the same ringtail, and this is I believe this is Rosie up here. So a few more views of some ringtails. This guy on the upper right was neat because we were doing radio telemetry, and I had my crews, and we had multiple stations. I'll show you how that's done. And as we were getting the readings, I noticed all of a sudden Brian Brown, one of my guys that was doing the telemetry, starts on the radio and says, "Hey, there's a ringtail up here. It's a wild one. It's not tagged." So we went running up there, and we foregoed the data at that moment, and we ran up there, and this guy just hung around and looked at us and was very curious. And we've had similar kinds of incidents like this in which Rosie Vizina says, "Hey, here's a ringtail here," and it just laid down on the branch, and we sat about 10 feet away from it quietly, and it watched, or she watched us while we watched the ringtail, and then finally the ringtail was like, "Okay, I'm satisfied," and went off on his own. So this is a young ringtail on the bottom right. You see the face is very, very much, with that young look to it. Some more ringtails. This guy's being held because it's tranquilized, and it's a female, and you can see the zygomatic arch, which is this difference between the cheekbones, is much more narrow on females versus adult males are much wider. So this is a male here, and he's got a much wider zygomatic arch. So if you have a mature individual, you can sort of tell what gender they are, but otherwise it can be a little bit difficult. And I noticed also the pigmentation on the chin and a little bit around the nose tends to be different. I mean, it's like a signature. So I started taking pictures of the chin and of the nose area because sometimes the ear tags will actually rip out. And then it's like, well, who is this? And then I'll compare it with the chin pictures. And it's like, oh, we got Ben, or whoever else it is. And when I mentioned that The underside of the tail tends to have a lot of white on it. This is a good view of the underside of the tail on the ringtail. Now, the area that I studied them was here in the Central Valley, and this is a Sutter Butte. So you can see that circular structure that I talked about that's volcanic. Here's Yuba City. So we're about 45 miles north of Sacramento. And this is a series of flows and ramparts, and you have the actual volcanic material that's been exposed from that volcanic plug that's come up. And there's some other areas like up here around Grimes, you have an area that's been pushed up by the valley floor, but it actually didn't break the surface. It didn't actually go through the valley floor and establish as a physical feature. Worldwide, ringtails occur. If you were to look at the continental United States and divide it into quadrants, so you have fourths, the southwest quadrant, all the way down to like the far western portion of Arkansas and up into Oklahoma, Texas, and so on, all the way down to Chiapas, Mexico, is where we have ringtails. Now, ringtails also have a Central American ringtail, which is called Bassriscus sumichrosti, and they occur in the deep, dense jungles, and they are very much more arboreal than our ringtail, which is named Bassriscus astutus. And if you know anything about Latin, and a lot of you guys are biologists, a lot of you folks out there are biologists, and you know that we use Latin because it's a dead language, so it doesn't change. So it has meaning to it as well. And the meaning of it for Bassriscus astutus, astute, is clever little fox, because it has a lot of that fox-like look associated with them. So this is a study area that I worked in. It's a crenellated, castellated core. Brian Hausbach over at CSU Sacramento has done a lot of work out here. It's about 10 miles in diameter, erupted 1.5 to 2.5 million years ago, covers around 46,000 acres. Now here's the Dean Ranch. This is this whole area right here is the Dean Ranch, and this is the area that I do most of my work. I've done a little bit of work with ringtails in Peace Valley as well, and then around South Butte was where I first started doing some work with ringtails with Gene Trapp when I started assisting him. So the habitats we have out here is more of a, it's a pretty dry environment. It's a chaparral environment. So you have lots of coffeeberry, one of my favorite plants. I really love coffeeberry. Toyon are in some of the lower areas, and especially underneath the trees. And then we have manzanita, blue oak, interior live oak woodland, with some of the lower elevations having valley oak in the areas with lots of sediment material in which water can actually build up, and lots of annual grassland. And of course, a lot of rock outcrops are associated with this as well. So here's some of those rock outcrops, and ringtails will heavily utilize those. Here's a good view of how some of the trees are scattered, and then you have grassland, annual grassland habitat, and some perennial species too of grasses like our state grasses there, the purple needle grass. And here we have some of the rock outcrops. You can see them extending all the way up. It's a combination of rhyolite and andesite that we have at this site. Forms some of these really interesting column structures that are huge. I mean, this is a very tall mountain here, and the fact that we have a couple of peaks that are or a peak that exceeds 2,000 feet above the base elevation of the valley, and then we have another one that approaches that too, qualifies this in many eyes to a mountain range. So essentially, we have here in the Central Valley the world's smallest mountain range, which is the Sutter Buttes. Let's see, let's go to the next one. So here's some more view of how the Sutter Buttes looks. This is from the side of that hill of North Butte that we were just looking at. And you can see on the north side of these hills, you get a lot more vegetation because those are the cooler sides. I mean, the only time we have the sun directly overhead is June 21st. And then from then on, it's always towards the southern, towards the south, and as a result they get less direct sunlight except on June 21st, and hence it remains cooler and a little bit wetter, so more vegetation can survive. And obviously these places are areas in which we do have good numbers of ringtails, and that's one thing that makes the Sutter Buttes so fascinating to me, is the sheer number of ringtails that we have out here. So the initial objective when I first started this was to determine home ranges for ringtails in the Sutter Buttes. And this actually started because the landowners of the sites, I've been going out there and doing morphometrics where we've been trapping ringtails, and I've been doing that as part of my class for the field ecology program. And I was helping Gene throughout the years, and we have lots of morphometrics on them, which are the body measurements. But landowners came up and said, have you thought about doing a home range study? I'm like, I would love to. They said, here you go, here's the keys. And that started it. And in 2008, I gathered up a group of some of my students that were willing to commit for a several month study, about 3-month study in the wintertime. So December, January, February is when we did this work, and started looking at home ranges. I was able to acquire 2 radio telemetry collars and enough receivers. I had to borrow one from USGS down in the Bay Area, but I was able to get enough that were on that frequency we needed. And we did 1 male and one female. Now, when we initially did this, it started off with these 2 ringtails, and the home ranges came out tiny. I mean, so small that I thought I'd messed up. And I thought I had messed up doing the calibration of sizing of the individual squares associated with the aerial photograph. So I recalculated it, came up same number. And I thought, okay, I'm starting to think now that I did mess up, and I did it a 3rd time, came up with the same number. I actually even went in and planimetered using a hand planimeter, one of those old style ones you could buy on eBay, but you can't find anywhere else. And it came out same number. So at that point I thought, okay, maybe I got some unusual ringtails. So I asked my crew, I said, you want to do this again? A couple people dropped out. It was just too darn cold and they were freezing. We got into the habit of providing a thermos of hot cider and a thermos of hot cocoa for my crew. And we were taking readings every 25 minutes or so. So eventually what happened was we ended up with 11 home ranges because we continued through 2010. So after the initial objective was determining these home ranges, and guess what? All of the other 9 additional ringtails we did had these tiny home ranges. So a supplemental objective came out. This is the thing about the science. You maybe haven't answered something, but then that just brings up 2 or 3 more questions, which I love, okay? And it ended up as, oh, well, why? Because determining the potential reason for small ringtail home range sizes, how can we do that? Well, let's look at what they're eating. Determine home ranges in areas with different habitat characteristics is something else that we might want to do. Because what's so special about this area? Because obviously the ringtails are able to get enough food to survive during the course of a year. Because we were catching them over multiple years oftentimes too. So after that potential objective, I started looking into a couple of things about home range. I found this beautiful quote by Gittleman and Harvey. They said in the carnivora, home range size is a function of the metabolic needs of a species, essentially what they are eating. So I started looking at ringtail food habits, and we ended up spending the full year of 2010 collecting scat and the poops of ringtails, and we ended up getting 461 of them and analyzed those. So the initial work started off in 2008 and 2010 and expanded from that first 2 ringtails as a radio telemetry study to 11 home ranges. Now, subsequent to that in 2010, we also added the food habits component into there because the question was, why are the ringtails doing this? So subsequently, in 2013, 2015, we did radio telemetry study on 5 home ranges on home range estimates in an area that is quite a bit different than that first area. And in particular, it was quite a bit different because there was less abundance of a particular species of plant that turned out to be incredibly important for ringtails, which was a stunner. We had no idea, and I'll get to that in a bit too. So what did we do? We live capture ringtails. This is a 6-inch Tomahawk live trap. It's covered with plastic material in case it rains, and it gives some shelter to the ringtail. And the ringtails typically drag that into their cage and make it into, oops, how did I lose that? There we go. Make it into a little bit of a den. And here's a ringtail that's actually in this live trap. Now, sometimes getting up to these areas can be a bit of a challenge. I was always deathly frightened about what might happen to the students, and I didn't want anyone getting hurt. And we had the usual, shin hamburgers and other things that happen at the site, but luckily no one got particularly hurt. And these rocks do roll a bit. So again, we're climbing up all around this area and all around this zone here that has a lot of dense vegetation. We were capturing ringtails in another valley, a small stream valley that's located to the left of this picture. A lot of it's dry habitat, and yes, ringtails will use this too. They'll actually use this as a highway by running along tops, but this happens to be that 2013-2015 study area. So it's quite a bit different. Going back to that first one, you can see there's a lot of rock outcrops and other things too, but there's a lot less rock outcrops on these hills. But the ringtails were still using this habitat, and most of these trees are blue oaks instead of the interior live oak and valley oaks in the lower elevations, in the lower valleys. So once we catch the ringtails, we tranquilize them. And I use ketamine hydrochloride. For those of you that are in the raving crowd, you know about ketamine as special K, and it is an illicit drug which makes you really high. And when you see the ringtails, oh yeah, they're high. And the beauty about ketamine, I have never had any ringtails go into seizures. And I had an interesting encounter with a graduate student out at Utah State University, and she was working on ringtails, and she contacted me and said, I'm thinking about doing something different because the biologist who I'm answering to in the federal government has said, give the ringtails xylazine. And when she gave him xylazine, out of 9 ringtails, 3 of them ended up going into full body grand mal seizures. And I've never had one like that. And I've tranquilized 200 or more of these. And I said, oh my God, you shouldn't be using xylazine. Why are you guys using xylazine? And when she got an explanation from the biologist said, well, that's what they use for cats. And one of the common names, it's a misname, is ringtail cat. They are not cats. They are not in the Felidae family. They're in the Procyon family. So they're obviously going to have different reactions. And xylazine is not the one to do, especially if you get a third of your tranquilizing subjects go into grand mal seizures. So I wrote this big long letter. It had 3 pages to it, giving my data. And that convinced them to allow her to use ketamine, and she didn't have any problems after that. But it allows them to go down for about 20 minutes. And, ketamine's sort of a happy drug, so they don't come out of it angry or mean. They're like, wow, what a trip I just went on. And they stay nice and placid. They'll do a little bit of salivation, so we try to give them back some supplemental water. And we put them back in the cage, let them recover for the trap, and we let them recover for about an hour before we release them, and then they're perfectly fine. So, talk about high ringtails. Look at these guys, okay? So these are a couple of ringtails that are tranquilized, and they're both females. And look at this beautiful apricot color you'll get on some of them undersides, and the beautiful tranquil liquidy eyes. And I wax poetic about ringtails, but they still remain as my favorite animal, and I can't imagine anything more charismatic. We've also noticed this as they get older, they start getting whiter whiskers, like a lot of us, okay, start getting a little bit whiter whiskers. And here's a cool thing, 5 toes. So most cats, unless they're polydactyl, are going to have 4 toes. And then notice that you have this main pad, which is just like what a cat has, but the ringtails have this additional pad. So if you look for tracks, and I was doing a project along the Kauai River doing a bat survey, and we found these tracks along Kauai River, which is awesome because we ran down to the hardware store and got some plaster and made some plaster casts of the tracks, but we had 5 toes with no toenails being shown. See, no, there's no toenails because they're retractable claws. If you were to take a cat's paw and push on the pads, the claws will come out. They'll just pivot out. Same way with ringtails. Okay, ringtails do the same type of thing. Okay, Let's see if I can get this to the next slide. So telemetry collars. I started off using a company called AVM, but I eventually moved over to Hollow Hill, and I've liked both sets of collars. I find that the Hollow Hill ones last longer. I mean, I can get 2 to 3 years on Hollow Hills. They weigh about 20 to 25 grams in body weight, so about 2% of their body weight. That way it's not encumbering them. This is one of the newer collars. You can see a little antenna. It's a little flexible whip. Ringtails will grab this and turn this around and around and around, and they'll try to get it over their head. So you have to be very cognizant about how tight you make it. It's not too tight, but it's not too loose, because they could get a paw stuck underneath there, and you can have lots of problems that can actually develop. After you do this, then it's time for telemetry. You can see this is one of the cold days, and this is a gathering spot, and then we move to each of the stations as we are connected by radio. And this is the Yagi 3-element antenna with these little walkie-talkie based telemetry devices. And once you get the data, then that's when you can actually triangulate it. But what we're doing is we're taking known stations. I'll show you this diagram in a second, and we get the bearing by compass and we write those down. So after we get the ringtail and put a collar on it, this ringtail has a collar on it, or we band it. This is a slow motion video of a ringtail that's leaving the trap. This is Scott Newton, one of my guys that's worked extensively with me with ringtails, and this is Raina. I believe this is Raina. Raina is taking off with the collar. Just lithe body and able to move very quickly through this kind of growth. So here are known stations, these red dots. These are stations that we've got affixed on a photograph. And what was in the past, we used to have an actual aerial photograph, and then we would log our bearings on that. But in the era of Google Earth, this is fantastic, because I can call this up and I have all of these red dots already set in there. And then I would use whiteboard pens, and I would snap on the grid lines for this because we were basing it on the grid lines for latitude, longitude. And we would take the bearing, I would make a notation over here, notation, 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 and then draw the lines, and where they intersect is where the animal's located. So it's very easy if you only get 3 readings to get a chance where one of those readings is off, and then that's no way you can use that data because it's not very, very accurate in terms of where that animal is. So we were getting upwards of as many as 8 bearings, and that gave us a really good ability to get some good data, because we always had plenty of intersecting lines. Now, obviously, you'll get ones that are going off in another direction because there's all these rock formations here. And if the ringtail is turned or something else is going on, it could actually reflect the signal off of large rocks. And then it sets you off by a couple of degrees or maybe even 20 degrees. So you can't use those. So the key thing is within a 5-minute time frame, get as many readings as you can and then log those. And then we wait 20 minutes between the start and stop point, and then we would take another reading. So about every 25 minutes, we would get a location data on the ringtail. And what happened was the 2008 through 2010 studies, we ended up with 11 home ranges. So these are all the home ranges. Different colors represent the area that was being utilized by an animal. Now, what's the difference between a home range and a territory? A territory is a defended area that is being the location, the area that is wandered by an animal during a particular period of time. Now, it can change by time. So we tended to limit all of our work within a 3-month time frame, because that's sort of a snapshot of what's going on with these animals. And as I record those bearings, as I input them into the program that analyzes these home ranges, eventually what will happen is the amount of hectares being covered will plateau. And when it plateaus, that says the animal's wandering within this area with very little likelihood that you'll have something that gets beyond that area. So you've basically got a pretty good idea of their home range. Now, if I took like 15, readings and put it together, I'm only getting a partial home range. So what I found was 45 was the magic number, around 44, 45. If you got 44 or 45 readings, then anything beyond that is just gravy and gives you some additional data. And then I also don't take 100% of the readings, because you might have had an animal pursuing something like a wood rat and goes out of that home range, but then it comes back into its home range. So those are called outliers. And we do a 95% minimum convex polygon, in which 5% of the most outlier 5% of readings gets dropped out of the calculation. So you have a pretty good idea what the home range is this way. Now in 2013-15, we did these 5 animals. And this isn't a trick of angle. The photograph is straight, or the landmass is straight underneath the photograph as it was taken. So there's no angle of difference going on. These are truly bigger. You can see that in the visual aspect. So when we started looking at this, the 12 home ranges, there's actually 11 here, but one of them's the same animal. So 11 animals, 12 home ranges. The average is 3.65 hectare. That blew me away. Okay, when I saw that, I was like, my God, I did work on the riparian zone of the Feather River, and it was bigger than that, significantly bigger than that. And you would think riparian zone is going to be much richer in terms of food resources. But it is a fragmented habitat, okay? So that makes a big difference. This is a contiguous habitat. So this is an intact ecosystem, and the home ranges are tiny. So how about looking at what happened in 2013-15? These home ranges, the mean is 15.11. So we're talking 4 times bigger, roughly 4 times bigger. So we'll get back to this in a second. What was amazing was that first result of those 12 ringtails. So when I started looking at the other home range studies in the Central Valley, we have this located here in the Sutter Butte. That's this study. We have Carol Liebscher's study, which is in the Butte Sink, so it's in the riparian zones, but they're very narrow bands of riparian, and the ringtails really don't leave those going out into open areas. They stay in these narrow bands. My study from 1993 was also in a fairly narrow band of riparian called Abbott Lake Ecological Reserve along the Feather River. And then this is Mike Lacey's work at Bobolane Audubon Sanctuary, in which it's also a linear type of habitat. And when they did their work, so here's my Wyatt, this is Abbott Lake. You can see that dense vegetation there. Here's the Feather River right along here. You can see there's more dense vegetation. This is O'Connor Lakes. And we actually had a ringtail that went down this corridor, came all the way around and came to this site. So we did have evidence of dispersal. This is Mike Lacey's site, which is the Bobolane Audubon Sanctuary. And this is Carol Liebscher's. Look how narrow those areas are. So in their studies, Mike, who was the first one, 1983, found 2 males, 2 females, and they were averaging 9 and 8 hectares. Okay, so the males are in blue. And then my study had the males having 4 of them, having 16 hectares approximately, and 8 hectares for the females. So we had a little bit of sexual dimorphism going on here with the home ranges. And I'm not finding this for the most part with the ringtails in the Sutter Buttes. Carol did 8 females and found their home ranges around 6, so it's comparable to what I was finding in riparian and what Mike was finding in riparian. And then this study, 5 males at roughly 4, so they're slightly bigger, and at about 3 hectares for the females on 6 of them. So that was the 2008 through 2010, because that's the one that we were interested in. When you compare it to other home ranges of other studies, and here's the Central Valley ones, Wyatt, Lacey, and Liebscher, and then this study with the ringtails. Okay, this study from the Sutter Buttes right here. But if you look at callus in coniferous forests of northwestern California, I mean, he's sitting at about 125 hectares for females and 275 hectares roughly for males. Coniferous forests, though. Cook and Brody did their work on males up by the geysers in Calistoga and found home ranges roughly about 220 hectares. This is Atchison and Harbison in Texas, and they found home ranges under 50 hectares. Yarchin in northern Arizona in riparian zone found home ranges that are pretty small again, 25 or so hectares. And then this is Gene Trapp's work out in Utah, which found home ranges approaching right around 125 and a little bit more hectares. This study, again, 3.65 hectares. So that raised a lot of eyebrows. And I knew that I had to have some sort of reasoning behind this. So it allowed me to delve into that some more. So looking overall, in the carnivora, home range size is a function of the metabolic needs of a species, Gittleman and Harvey. So looking at this, let's look at Scat, because that's what we started collecting. And this is actually freshly deposited. If you could see it, this is actually still steaming. And I got to tell you, the great story about this is this guy here on the left, right here, Michael Starkey. He was part of my crew. And while we were doing telemetry, all of a sudden a ringtail shows up in the tree that's in front of me. And Mike was just up the hill from me. So I called him over in the radio. I said, Michael, I have a ringtail in the tree in front of me. So he came down and we looked at it, and we had just been talking about feral pigs in the area and how sometimes people just find their dentures or something because the feral pigs have eaten them. So we were sort of wigged out about feral pigs because we could hear them down below. And all of a sudden, Sarah shows up right over our shoulders. We didn't even know she was approaching. She goes, "Hi, guys," and we almost fell down the hill. We were so startled. But we ended up watching this ringtail for a little while. And then the ringtail comes down the tree, and it went through this dry grass without making a sound because it would run along limbs or logs that were fallen on the ground, would jump between those and rocks, and would actually not make a sound as it traveled. And Michael started running alongside it to watch it, from a distance. And then this is when I knew I had Mike as a scientist, that he was going to be a really good scientist. It decided to sit here and take a dump, and it pooped. And Michael is screaming, "It's defecating, it's defecating," and instead of pooping or crapping or something like that. I thought, oh yeah, here's a scientist for you. So that was fun. And he is a scientist. He runs Save the Snakes, which is a semi-nonprofit that does a lot of snake work worldwide, and they have affiliates in places like India and in Africa. So he's got a great organization. But here's a good example of a scat. Now this isn't a ringtail scat, because ringtail scats are much smaller. This is probably a fox scat. And this is Brian Brown, who was one of my folks too. We collected 461 scats in 2010. I'll show you where we collected them, and 400 scats in 2015. So looking at what other species we had out there, I put out a bunch of camera traps. We figured out what might be something that we could mistake it for. Well, we had raccoons. Raccoons are huge. They're about the size of your thumb, much bigger than a ringtail scat. They have different morphology. They break off in abrupt ends and so on. Striped skunk would give ringtail size scat, but they also have a characteristic little aroma with them. And there's also some differences on it physiologically or physically that allow you to discern differences. And then gray fox. Now, a small gray fox can give a ringtail sized poop. And ringtail sized poop is about the size of your pinky for a decent sized one, maybe even down to about half the size of the pinky. And they have a particular taper that's associated with the muscles around their rectum that cause a nice narrowing of the taper, versus a fox tends to have a little bit different morphological structure. But the big difference is canids have nasty anal glands, and they produce a very, very nasty smell. So one of the things we do is we take a sniff of them. And if it was like, oh, it's terrible, we didn't collect it. And if it was too big, we didn't collect it. We collected anything that was about the size of your pinky and pass the sniff test. And then we would bag it, put it into a paper bag. As you can see from the previous photo, go back to that. Here we're putting it into a bag. We mark the GPS location of the collection. We mark the date, who collected it, and oftentimes what kind of habitat it was in. So that's how we were able to tell the difference between ringtail scat. And then when we get it back into the lab, they're dried, so we would put them into beakers with hot water, enough that would kill bacteria and other potential parasites, and stir it up with glass rods, and then we would strain it through very, very fine mesh strain, collect the leftover portions and then put them onto a Petri dish and look at what they contain. And this is a good view of Rhamnus ilicifolia seeds. This is Microtus californicus hair. So we would take the hair and make a slide out of it. And also we would do a sniff test here too. If we got a foul smell, then we would just dump that because there was a chance that it was a gray fox. So true, it would have been better if we had done DNA on them, but hey, I don't get any funding for this stuff, so $25 a sample wasn't going to happen, not with 461 samples. So we decided to rely on multiple methods to ensure that we got ringtail scat, and we're very confident they were ringtail scat. Dominant food items, a lot of plants, oak mistletoe. This is the unusual one. And here's the berries. Yes, I tried the berries myself because ringtails were eating it. It's a little astringent, but no bellyache or anything. This is hollyleaf redberry or evergreen buckthorn. This is the Rhamnus ilicifolia. Lots and lots of these delicious little berries on them. Coffee berry, and they produce these huge seeds, and they're discontinuous ripening, just like with the mistletoe. So these guys will produce coffee berries over the course of about 3 months or so. So that's good. And then toyon, similar type situation. Toyon stays put. And in fact, toyon dries on the branch, and they're still quite edible as it dried. It's sort of like eating a raisin. And then, of course, insects. They were hammering those guys too. So looking overall at the food items that were consumed, the dominant ones, the main ones, were oak mistletoe fruit, oak mistletoe leaves. And I'm going to come back to this in a second because this is what blew me away. California coffeeberry fruit, so they're eating the fruit. Hollyleaf redberry fruit, so remember here we have mistletoe leaves too, but toyon fruit, and then blackberry fruit. So there were some non-native blackberries in the area, and they would actually consume those. And oak acorn for the course of about 2 months. The species we have of oak mistletoe is Phoradendron villosum. So that was the specific species we're looking at. Now, as far as animals are concerned, there's a lot of invertebrates. Beetles and beetle larvae. It was interesting, the beetle larvae were being consumed, but we also had lots of noctuid moth larvae, which is the butterfly family, or butterfly order. And found out that noctuids will overwinter as caterpillars, and they will oftentimes form into these large aggregations where they could basically conserve heat. And during the really cold nights, they could vibrate a little bit, producing enough heat that they survive. Well, you know what? Ringtails were finding them because every time we found noctuid larvae, we didn't find 1, 2, or 3. We found a big mass of them in the poop. Grasshoppers and crickets, lots of Jerusalem crickets were being eaten. And then seasonally, things like field crickets, and then during the summers, grasshoppers. Dragonflies and damselflies. So they would encounter dragonflies and damselflies that were roosting for night, and they would consume them. So they're opportunistic. Mammals, in the Sutter buttes, mostly they were the deer mice group, the Paramiscus. You can't tell the difference really well between the 2 species of deer mice that we had out there, the brush mouse and the deer mice. So we just kept it at Paramiscus. Species, and then we also had Microtus. And then birds, mostly we would get these in the spring. And reptiles, we had a few reptiles. We had some lizard, and we had some gopher snake. And then centipedes and millipedes. And the millipedes were a little bit of a surprise because they produce a cyanide-like compound that'll make you sick, but they were also limited in terms of what time of year they ate those. And as far as the centipedes, it was everything from those really large, big, blue-legged guys all the way down to the small soil centipedes. So looking at the frequency of food items, look at these numbers. Here in September, 100% of the scat, that means 55 out of 55 scat, contain plant material. 97%, 95, 97%, 97, 95, 70% in January. So it's pretty obvious that plant materials was a very significant component of their diet. And then look at insects. Insects are also big. April is when you're going to have lots and lots of insects that are available, so 97%. And then, of course, in the winter times when it goes down. Mammals, relatively significant component during the early part of the year. Not much so in the summer, and then again reappears a little bit in the fall. Birds, spikes in the spring. So here we have February, March, April, May, and then it stays steady, and they're catching things. I mean, we found scrub jay feet, robin feet and feathers that we could tell what they are because of the feathers too, and we see them in the winter time. Reptiles, a little bit in the summer, a little bit in the fall, but not much else. Chilopoda are going to be the centipedes, and we see those present. And of course, this is a 2010 results, and then look at this number: 35% of the scat contain millipedes, and 17% of the scat contain millipedes in April and May. So is there something that goes on after the spring? Well, think about this. Millipedes are herbivorous. They eat vegetation. April is when you're getting some of the rapid growth. You're getting the spring showers. So does that water content reduce the concentration of whatever toxins that they produce? I don't know. That would be a really fascinating thing to actually study. So again, see, I told you about how biology leads to more questions every time you might answer a few. So I love this graphic because the blue shows you plants. The reddish coloration shows you insects. Look how significant these two items are throughout the course of the year. This is January on the bottom, December on the top. I wish it flipped over, but it didn't go that way. This is birds on the purple. So birds form a pretty steady amount of protein material. You have mammals that also appear as protein material, but mainly the proteins are coming from right here, the insects. And what's going to be interesting is some plants produce quite a bit of protein, and then very little in terms of the others. Again, this is the 2010 data because that's the stuff that correlates to those really small home ranges. So when you graph them out in this three-dimensional graph, there's always something for them to eat. Notice that there's always something. Here in January, they're mainly feeding on mistletoe berries, and you got both berries and leaves coming in. You also have insects here. So you have the beetles are present. And then it continues on, starts to drop off, and you have a replacement with the Rhamnosalicifolia right here. And then this lighter purple is grasshoppers, crickets. And then you have the coffee berry coming in play. You have mistletoe berries again. You have, this is the acorns. So there's always a constant level of food resources that are available for them in that particular area with the small home ranges. So looking at these ranks in January, oak mistletoe fruit was #1 thing, and then beetles and beetle larvae, and then mammals, and then you have February oak mistletoe again, but the leaves start coming in. Oak mistletoe, notice through April, and then oak mistletoe is right here, the second most prevalent item in May, with crickets and grasshoppers starting to show up. And grasshoppers and crickets dominate, and Rhamnosalicifolia, and pretty much mistletoe drops off. Going to the next series of months, here you have that Rhamnosalicifolia being prevalent. Then you have coffee berry being prevalent, and you see Rhamnosalicifolia, they're eating the raisins basically, and you have lots of insects, and then acorns start showing up. And then acorns jump to being the #1 items for November and December, with oak mistletoe coming in a second, and some of the toyon coming in right here too, but with also birds showing up. So these are just sort of the ranks in terms of what was prevalent. Now going back to the mistletoe, all that resulted in, quite frankly, I thought mistletoe was not a good thing to have. I'm of a different mind now. Now, invasive mistletoes are a different matter. But I found this paper by David Watson, and it's from 2001. He started looking at native mistletoes. And they have a 4 to 6 month flowering and fruiting season. So the fruit are available for up to half the year. The fruit are 40 to 60% dry weight of soluble carbohydrates, quick energy, carbs. And the fruits also have protein averaging 20% and have 18 of the 20 amino acids, including all 10 of the essential amino acids. Wow, I had no idea. Fruits also contain high concentrations of potassium and phosphates and micronutrients like manganese and iron. Toxicity is rare with the native mistletoes in general, and the discontinuous ripening of fruit results in ripe fruits being available throughout that fruiting season. And this paper is phenomenal because it talks about how many species depend upon mistletoe. And then I looked also, the Ernest wrote this report in 1969 in Arizona, found that crude protein levels of desert mistletoe was ranking from 7 to 25%. And all mistletoes examined contained more moisture than their hosts. Ooh, desert environment, moisture. We're sort of a desert environment with the Sutter Buttes. For deer, in terms of digestibility, mistletoe is considered excellent forage. I've heard people talk about how if oak mistletoe gets thrown on the ground, the deer are all over it right away. So you don't see it very often on the ground because the deer consume it. Mistletoes, when available, may supply sufficient energy to reduce the need to metabolize protein for energy because it's a vegetable protein, thereby releasing dietary protein for tissue maintenance and growth. Something else that's cool, doesn't fight back either like mammals or birds would. Here, Hull and Leonard in 1964 found Phorodendron, mistletoe family, that's the genus that includes the oak mistletoe, essentially a water parasite and not a nutritional parasite of the host. So under small numbers, they're actually not bad for the trees. Phorodendron has only occasionally been noted to cause injury to either ornamental or forest trees. So again, this whole idea of, oh my God, mistletoes are terrible, this sort of turns it on its ear. So looking at mistletoe frequency of occurrence and relative frequency. So what frequency of occurrence is what percentage of scats contained that. And then relative frequency is what percentage of all the food items did it comprise. So because you don't just have one food item in scats all the time, you can have multiple food items, you're going to have a lot larger number in that situation. So looking at frequency of occurrence, you see fruit is dominant both here in November, December, and January. Fruit is the biggie. And then in February, it comprises pretty even numbers almost between fruits and leaves. And then in March, it switches 94, 92%. Why? Because the berries are almost gone, completely gone during that time. And then you see it coming back a little bit more here. And then June, you see mostly leaves. And then look at the zeros all the way across and near zeros across here. In terms of relative frequency, what percentage of the diet? 29 and 10, 20 and 17. So you see it's 39% of all of the food items. Oh, crap. There we go. Of all the food items, 36%, 31%, 29%. Then it drops down to 8% of all the food, 1, 0, 0, 2, 19, and 20. It's very important for their diet. So what about mistletoe then? That got me started on mistletoe. Everyone knows about mistletoe around Christmas time, right? Sneak a kiss if you can with that. But in terms of nature, here's how we'll oftentimes see the mistletoe in these large clusters. And these are the berries, and these are these succulent kind of leaves, and they got this viscin, this mucilaginous material that in the case of birds, they distribute them on trees because it's sticky, and when they defecate it, it sticks to their vent feathers, and then they rub their vent and their butts on the tree limbs, which then forces the seeds into the crevices, and then they develop their root system, which is called hysteria, and it invades the tree and then invades the cambium layer, and they start taking water from the xylem and food from the phloem, and that's how they end up surviving. But mostly it's xylem tissue that they invade. So is it a blessing or is it a bane? You'll see a lot of articles about mistletoe, but most of it's the European mistletoe that is a bane to a lot of trees, or some of the mistletoe associated with cottonwoods and the similar type of species, and they could be pretty detrimental and bad. But overall, oak mistletoe and desert mistletoe, it's not a bad deal. They actually are something that provides quite a bit of resources for organisms. Now, of course, if you have this infestation, something's odd with these trees causing this kind of growth, yeah, it's going to damage the trees and cause a lot of issues. But for the most part, this is not what you see. Normally you see 1, 2, 3 clusters of this, and that's it. So with the ringtails and this paper, what I came to realize very quickly was that yes, it is beneficial. Look at the prevalence of mistletoe in terms of the fruit and in terms of the leaves. It's significant except for these middle months. And if you look at these middle months here, we have Rhamna salicifolia in blue. We have Frangula californica, which is the coffee berry berries, and then we have acorns. And doesn't that correspond to time in which this mistletoe is not available? And look at this, mammals and birds, which is a smaller component of their diet, but provides a big protein boost, supplements the protein aspect to it. And if you look at beetles and grasshopper family or order, they also fill in during these periods of the time of year in which you have less vegetation resources available for them. So that's that whole graphic that shows the year-long feeding that they're able to do on these small home ranges. So wherever an organism is provided with resources that allow it to survive, that's its habitat. And here's the overview where you see they have plenty of food resources that they can take advantage of during the course of the year. So yeah, mistletoe is a blessing, okay? It's actually good. You know, Bill and Ted's coming out with a new movie, so I had to put this in. So much older Bill and Ted. So in this habitat of the Sutter Buttes, okay, we have this habitat along North Butte, which was the 2008 through 10 study. This is North Butte looking towards the north, and this is looking towards the south from North Butte. Turns out that that's going to be something important. So what we did was we went out and I sent out teams of my students, 2 people, 1 person with a GPS unit, another person with a data sheet, and then with chalk. And we just put an X on the tree's trunk, and we would log what species it was, and if it had mistletoe, and how many clusters of mistletoe. What we found was these are the areas which had mistletoe. And when you break this down, the total number of trees, 980 interior live oak were counted, of which 450 had mistletoe. That's 46%. Blue oaks, 425 of them, with 93 of them had it, so it's 22%. And then the valley oak, there were only 40 of them, but 18 of them had it, which is 45%. That's a huge rate. So this was curious to me. And in talking with Jenna Meyer and other botanists, turns out blue oak has a much harder, much denser wood. So it might be more difficult for mistletoe to actually invade it. So you have a smaller number. But the vast majority of them were interior live oak. So what that did was it led me in 2015 to this area. And notice this area is the area I collected the 400 scats. And what I found in the 400 scats is this is the blue number. That's the relative prevalence of mistletoe fruit and leaf in comparison to the 2010 results. But here's a cool thing. Almost all of this area, which is the area that we had the other home ranges, is almost entirely blue oak. And that there are patches of interior live oak, and then even lower numbers, only a few handful of valley oak. But the blue oak sat at about roughly about 5% rate of having mistletoe. So mistletoe is a little bit more difficult to get to, but the numbers don't reflect that 5% and 9 times less mistletoe. You would expect these blue bands to be smaller. So the ringtails still are seeking out mistletoe. When they do find it, they eat quite a bit of it, and it shows up in their scat. But they were mainly subsisting also on a wider variety of food resources and a lot of insects. They were eating a lot of insects in this one area that's outlined in yellow. So we did not have these kinds of numbers like we had at that first area. And then we also have home ranges that are 4 times bigger. So I think that's the big issue. That's the big thing that's causing those home ranges to be so much smaller at that one area. So keystone resource for ringtails, I'd say absolutely. Frequency of Pacific mistletoe can be high in some areas like that 2008-2010 area, but it remains relatively uncommon in terms of relative size of the resource. Pacific mistletoe is a major component of ringtail diets during periods with few other vegetation resources like other fruit. The presence of Pacific mistletoe reduces the need for a larger foraging area for ringtails suggested by the larger home ranges in areas with very low frequency of mistletoe. So there's a lot of circumstantial evidence, strong circumstantial evidence about this. So is it a keystone species? Well, Watson describes 97 vertebrate families that consume mistletoes worldwide. 97 vertebrate families, with another 50 vertebrate families using mistletoe's nesting sites, birds, and mice and other things. Most species of mistletoe are dispersed by animals, chiefly birds, and mistletoe and birds are considered a model system in terms of fruit dispersal. And you get things like phainopeplas, which depend absolutely on that mistletoe. There are a large number of insect species that depend on that mistletoe, with some of the large purple hair streaks or skippers that will actually be found only associated with mistletoe. So the conclusion were that metabolic needs of these ringtails are met on these small home range sizes in the Sutter Buttes, and mainly it's because of the mistletoe presence. The 2008 to 10 home ranges are the smallest documented home ranges for ringtails. Sequential availability of food items occurs in this area, which allows them not have to travel very far. Imagine if just one or two or three of those items weren't present. They'd have to wander a larger area in order to get enough food. But because we have an intact ecosystem, they don't have to do it. So this is actually another argument for intact ecosystems. It makes me wonder, prior to human settlement, or I should say European settlement of California, what must have been the distribution of ringtails? Because, we only have 5% of the old growth riparian forest left in the Central Valley streams. And we have about another 5% growing back, but we used to have 1 million acres of that, and we're down to 50,000 acres of old growth. What was it like, and what kind of travel ways did those provide for animals like ringtails and bobcats and raccoons and all of these mesocarnivores, and even the big mega carnivores like mountain lions? What did they travel? Mistletoe is an important means to meet ringtail metabolic needs, and insects provide also a reliable food source throughout the course of the year because you have things like Jerusalem crickets available through the winter months. So my crew, okay, I have to name off, and there's more people than this, but these are the people that stuck it out on those cold nights and freezing and doing things over multiple nights. Kathy Norton, Michael Starkey, Sarah West, Scott Newton, Jamie Gergany, Stephen Gergany, Katie Rush, Brian Brown, Shauna Jenkins, Rosalinda Vizina, Michael Mamola, Christopher Vang, Brijesh Varma, Tina Favela, Brian Cornely, Raquel Elander, Amelia Tauber, and the many others that spent 1, 2, 3, or 4 days out there helping out with things, and they got as their reward their ability to hold a ringtail. Tranquilized, of course. You don't want to do this to a non-tranquilized one, but they are an amazing animal. Actually, we don't have a picture of Brijesh with a ringtail, do we? Okay, he's got a snake. Ringtails eat snakes, so that's good too. Special thanks to Margaret and Peace Sands for giving me the opportunity to go out to their property and do this work, and always being so supportive of this work. And I love you guys dearly. And of course, Gene Trapp, you introduced me to ringtails. You made me fall in love with them. Actually, it didn't take much making to do that. And you've been my sounding board for years and years, and one of my closest friends. So thank you for that. And then to the nocturnal subjects, the ringtails themselves. I can't imagine a better subject to study and to devote a lifetime to actually learning about. So thank you to those guys. A little bit of strawberry jam and cat food may not seem like much of a reward, but they are willing to take it as pay, so that's good. So some pictures of ringtails to end it out, close it out. This is Ben. After release, he went up into the tree, and we usually watch them for a little while, make sure nobody like predators might get them, and they come down and they go scampering off. Before we do this, we always give them a little bit of carbohydrate boost. They love strawberry jam of good quality strawberries, not the kind like Rainbow Brand or something like that. They want Knott's, Mary Ellen's, or Smucker's, and it's got to be the preserves too. And here's Katie, and this is Jack, a couple of beautiful pictures that they were very cooperative about and did. Another ringtail, I can't remember who this one is. And this is a ringtail in Manzanita during a rainstorm. Again, it went up into the Manzanita. Releasing of a ringtail before it goes into vegetation. Now, of course, we wear gloves on these. Sometimes you saw pictures of us without gloves. That was sort of the old school way to do it. We're actually now doing I'm doing COVID protocols on any animal that I'm going to handle from now on. I'll be training that way, where we always wear gloves, we always wear some sort of face covering, and it's basically we don't want to transmit COVID to these guys. That's why I'm not doing any bat work right now in Cal Fish and Wildlife. It's pretty much suspended all bat work for permittees, and I want to do the same thing with ringtails. I think we have to be very much aware of reverse zoonosis, where we're going to provide. We're going to be the vectors to these animals, and also the animals could end up as vectors to us. So we have to be very cognizant of that whenever we work with mammals. Another picture of a ringtail. This is a beautiful little male. The tail is amazing. I hate seeing those tails on eBay because Texas, they still hunt them. This is a state mammal of Arizona too. So, and is a fully protected mammal here in the state of California. And this is the ringtail that was in the Manzanita. And this is a baby female, and she decided there's a den site. And this is what it looks like in the den site. And this is Sally on the bottom right. And this is her daughter. And she had a daughter and son that I discovered when she wasn't moving from a den site. I started looking in there, and I could see movement of something that was small and had ears that did not have their cartilage yet going. So the only way I could actually get a picture was I took my iPhone out of its case and jammed the iPhone and took some pictures using that, and then I was able to discern that there were babies in there and watch them for a little while. It was pretty amazing. Now, what was interesting was a month after that point, and the babies were probably still eyes closed, she moved the babies. And when I went over and checked the den site, I got covered in fleas. So what they'll do is, while they're in a den site and the babies aren't ready to move, they'll just sort of endure the fleas, and then they'll move to a new site when the fleas become bad, and they'll switch around den sites. I think that's why they move around so often. This is a video of Ben, and the reason I know it's Ben is because I put a little bit of reflective tape on his collar, and the one dot corresponds to Ben, and then other ringtails had different numbers of dots. So this is him coming over, and I love this video because what does he do? This is that den site where you saw those other ringtails. That's a toyon with berries, and watch him reach across. So he's listening, and this is a black infrared stealth cam, so he doesn't really see it, and now he's going to reach out and try to feed on that. Oh, this was so cool. It's just really validated to me that they are actively foraging on this stuff, that they're not just grabbing it off the ground or something like that, that they're actually feeding on it. So this is feeding on toyon. Now, out of all of my camera traps, ringtails are my #1 species that I get in the Sutter Buttes. And again, that's in the Sutter Buttes, and I think it's because it's an intact ecosystem and you have a very high density of ringtails. You don't have very many bobcats. You don't have mountain lion. You don't have wolves. You do have coyotes. You do have foxes, but not particularly many of those either. So I think their numbers are nice and strong. We'll also get lots of foxes. This is my other species that I'll get on my camera traps on a pretty regular basis, would be foxes. And the gray fox, and I've only had red foxes on very rare occasions, but that's the Sacramento Valley red fox that we have up here. And this is the gray. The grays have that black tip to the tail. Ironically, I've actually seen gray foxes crawl into that opening. I mean, they have to do the army crawl type of thing, but they actually do go into that. And there is a back door on this side to this, so if there's any animals in there, they do bail out in case foxes go after them. But most of the time, whenever we've seen any sort of fox ringtail encounter, they just look at each other and there's not much that goes wrong. Okay. Beautiful animal, the fox. And they also have retractable claws too, so they're excellent climbers. And as promised, I'm going to show you cuteness personified. This is at the Oregon Zoo, and this is a baby ringtail. It's only about a week old. And it is just so cute. You can see his ears are still a little bit floppy, so the cartilage hasn't fully developed. They had 4 babies born, so you could hear the other babies. I wish you could see the little stuffed raccoon that they have. It's a plush toy. That's the baby ringtail's companion. And Oregon Zoo has them. So does Sonora Desert Museum and outside Tucson, and Living Desert Museum out in Palm Springs area, Desert Springs. The Coyote Point Museum, now called Curiosity, used to have them too, and they don't have them any longer. All right, so that's the end of the talk. I'm going to actually come out of the screen and stop the share, and we can open it up for questions. I hope you're as captivated with ringtails as I certainly am. I suspect most of you guys do appreciate what they offer us in terms of beauty and in terms of their ecosystem function. All right, thank you, everybody. I'll turn it over to Bill.
Thank you very much, Dave. So there have been some questions that have been popping up as you've been talking. And do you want me to read through them for you, or do you want to take a look yourself?
Sure, I could see them right here. Let me start off from the very top. Let me go through them. Hi, Dave. Hi, Jesse. Okay, all this stuff. From Gene. Uh-oh. I understand that you're contributing information on ringtails in a book on carnivores. When will we see this book? That's a really good question. I think everything's on hiatus and on hold. There's a book that's coming out called Fur-Bearing Mammals of North America. And because ringtails are harvested in some states as a cheap $2.50 fur, which unfortunate, but they are being harvested. So I was asked to co-author a chapter on ringtails with my colleague from Utah State University. Oh my God, her name is, I just lost her name. Now that I'm thinking about it, I'm not going to be able to remember it. Anyway, this book was supposed to come out this summer, but I have not heard anything about it, have not seen any additional reviews. So that's a very good question, Gene. I hope it still does come out. I mean, it was in the final edit, so it should. And then Lauren DeBoer says they're nocturnal, but these are daytime pigs. Do they also come out in daytime? Very, 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 very rarely will they come out in the daytime. I've seen them sunning themselves out of a wood duck box, but they're not usually out. The reason they're out in the daytime is because I just live trap them. And because we work them up and then we release them, that gives the photo opportunity to be able to take pictures of them. A few of those pictures were nighttime. Those were the ones that we encountered actually at night, but that's a pretty rare occurrence. I mean, out of all of the years of doing radio telemetry out there, I've seen 3 ringtails. And I knew they were out there because I can radio telemetry them, but we never really did see them. Erlen says, my grandfather caught one in his attic. He was hunting bats up there at night. I named him Legolas because Lord of the Rings ringtail. That's pretty good. Yes, they'll get up into attics. They'll get into your houses. Gene Trapp was telling me this wonderful story about a guy named Durham Giuliani. Durham Giuliani, he had visited Durham, and Durham lived it was one of the creeks in the Bishop area and Lone Pine area, and he had a stovepipe which wasn't present, and a ringtail would come up that stovepipe and come into his house on pretty much a nightly basis. So he would sit real quietly and put food and raisins on his leg, and the ringtail would come over and take them. And I guess one of the first encounters he had, Durham had just gotten in from the field and hadn't had a chance to take a shower and knew the ringtail would be coming in. So Durham stayed really still, and Gene was telling me the ringtail came over and sniffed his legs and grabbed the raisins and ended up on his shoulder and was sniffing around, and Durham wasn't moving at all. And because he was smelly, ringtails have a keen, acute sense of smell, he went over and put his mouth on Durham's ear, and Durham still didn't move. So then the ringtail bit down, which made Durham jump, and then the ringtail scampered off and said, yeah, I guess he is alive. So he sort of smelled dead. And he had another experience in which Durham had a captive ringtail that was living in his station wagon. When Durham visited Gene, there was a ringtail in his station wagon. So yeah, they do get caught in buildings. There's a couple of places in the Big Sur coast, like these cabins that you can rent that have ringtails coming out regularly. And then out at Mojave National Preserve, out at the guy from UC Santa Cruz that established, it's called Ringtail Cabin, and he established a little field station out in the Granite Mountains, and ringtails come regularly into that if you leave the door open. So you do encounter them. And then also Yosemite, at one of the lodges, ringtails will be up in the attic or in the rafters. As you're eating dinner, you'll have ringtails moving around sometimes. So Michelle says, what are the main differences between ringtails and cacomissals? Well, cacomissal is the Native American word for ringtails. And the other thing too is technically cacomissal is a Central American ringtail, which is Bassriscus sumichrosti. So the same genus, but different species. And then cacomissals are sort of used as the Southwestern Native Americans use that as a name for them. Kirk, maybe you'll come up to this, but what is the definition of small home ranges? Well, small for a mesocarnivore is that 4 hectare. Okay, so we talk about that small home range, and it's small relative to the other home ranges which are documented. So it's a relative measure. Kirk, again, what you're reporting about the different tranquilizers effects seems very important. Have you happened to follow up with wildlife veterinarians, zoo veterinarians for understanding how broadly this difference in effect, mainly seizure risk, is understood previously observed for procyonids? Yes, I've had several wildlife veterinarians. I think Deanna Clifford was one of them that I was talking with at Fish and Wildlife. They would not use xylazine on procyonids. So this was a situation where a biologist was not familiar with procyonids or ringtails, and because of the misnomer of ringtail cat, suggested something that they found is usable in cats, and it isn't usable for ringtails. So I prepared a scientific poster and presented that out at the Wildlife Society meetings too. And I've had conversations with a number of zoo people, zoo folks from around the country. Michelle, I'm not sure what they use at work zoo, but we use ketamine for a lot of animals. I've never seen a seizure during any procedure over the years. Yeah, I love ketamine. I wish it wasn't so hard to get. I'm actually using expired ketamine that's been kept in a refrigerated condition. I just watch it real carefully for any flocculant, but that's the only way I can actually get it. Otherwise it's very, very difficult to get. It's almost like the Wild West with ketamine. They're actually having incidents where you're having semis that are carrying veterinary drugs get hijacked, and they just go in and they find the box with ketamine, and that's what they take. And it's like the Wild West in that regard. So it's very difficult to get, but it is also probably one of the safest drugs, I would say, that we have for tranquilizing animals. Lauren, do ringtails have natural predators? Yes, actually, Rosalinda Vizina and I, we actually had a ringtail, because the radio collars have the ability when they're not moving for 24 hours to basically double or triple the signal. So basically, instead of going beep, beep, beep on a particular frequency, they'll go beep, 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 beep. So you know it's not moving, and you're hoping the ringtail will drop the collar, but it could also be a mortality. So we followed the signal until we finally found it in this spot, which I would never have thought a ringtail would have been. It was one of these huge erratics, volcanic erratics, maybe about a 4-ton boulder that had been thrown out of the volcano and landed. It was sitting maybe about 3 feet off the ground and was about the size of a car. And it was coming from this rock, and it was all grassland around it. So I thought, what the heck is going on here? So maybe it's in a burrow. And I kept getting it from the south end of this rock was the strongest signal. I walked all the way around the rock. So then I took the antenna off and just used the cord and found the strongest signal was on the south end. So I took the cord off, and then I noticed the signal was coming from the south end. There was no evidence of a collar, no evidence of a ringtail. And I started thinking, wait. So I pushed the antenna connection up against the rock, and wham, the signal came on. And it was just like overpowering kind of signal. And I came to the realization that volcanic rocks have a lot of magnetite and have a lot of magnetic properties to them too. So I went to the other side of the rock, and lo and behold, there was disturbed soil. And as I dug down to the disturbed soil, about 4 inches down, I came across ringtail fur. And I excavated a ringtail that had been killed within 24 hours, and it was deposited in a cache. Now, typically canids will do this. So the suspect was a coyote. So I took the ringtail. It was in great shape, except for it being dead, and that's a bad thing. And coyotes got to eat too. So I took it and we froze it, and we ran a necropsy of it at California Department of Fish and Wildlife Region 2 with Deanna Clifford and Jamie Rudd and Rosalinda Paisina. So I watched it, and it turns out when we opened it up, on the left side, it had 5 broken ribs. It had a punctured lung. The teeth spacing, the canines, because we knew it was a carnivore then, the canines were spaced by 30 millimeters, which is ideal for adult coyote. It's the only thing it could have been. And we also noticed there was blood in the cerebral spinal fluid, and that would be with shaking, and there was shake damage post-mortem to the liver, where there was stretching but not bruising. All that was consistent with what canids do. And there was a canine puncture at the ventricle of the heart on the bottom, which filled the pericardium with blood, so it bled out almost immediately into its pericardium. And that was fascinating results. So that's something that I've got almost completely ready for publication, and coyote got it. But the other thing that will get it would be gray horned owls and hawks. And the gray horned owls don't have a beak structure like hawks and eagles do, where they actually dismember their prey. So what they'll do is they'll pretty much leave the body intact and take the meat off of it, like how we would sometimes eat a chicken, maybe, just taking the meat off of the chicken. And you'll find the fur peeled back around the wrists and around the face. And I found one of my ringtails, Don, got predated that way. So I'm assuming it's a gray horned owl that got it. So those things will actually get it. And Native Americans will sometimes, in the northwestern portion of California, they'll use them for ceremonial cloth. And the Mimbres, Native Americans from the Southwest, use them in pottery and stuff. And I'm just going to show this off if you can. This is a Mimbres pottery of ringtail. So it's over at the DeYoung Museum from 1150 AD. So that was my tattoo. Had to get a ringtail tattoo. Okay, Kirk, what time of year do ringtails raise young? They're going to be pregnant around April, and then into generally around June they'll have babies. The babies stick around through the fall. It seems like the females stay around longer with mom than the boys do, but then they disperse and they're ready to reproduce after a year. Do you suspect there may be a dietary change related to reproductive cycle needs? That's a really good question, because maybe later on or during the course of the year they need more protein for milk production. So that's a good question. We don't know. I actually will not live trap ringtails after April or after middle portion of April. I don't do any. And I will not resume it until usually around October or November. Mainly because I don't want to introduce ketamine into potential fetal development and into mother's milk. So I choose to regard the ringtail's health as number one beyond the data that I would get. So that's a question where DNA data would be interesting. Yes. And I think it'd be really cool to take fur data and look at some of the radioisotope materials there too, because that might tell us some information about the birds. What kind of migratory birds are they taking, for example? Sex-specific differences in diet choices in different seasons. Yeah, that would be fascinating. Is it possible the ringtail switch food items during gestation or when feeding young due to nutritional needs? And again, that's a great question. I don't know. I think that's some more questions to explore. And hey, if there's someone that wants to do grad work with them, I have the permit. You could get on my permit. David Rosen, riparian zones. Do ringtails eat the mistletoe that's prevalent in cottonwoods? So I did food habits analysis up along the Feather River. I did 272 ringtail scats, or I'm sorry, 172 ringtail scats, and there was no mistletoe present, even though there's mistletoe present there. My understanding is that mistletoe, the berries, will give you a nice stomach ache. So whatever is in the populus group is the stuff that might be more toxic. So maybe ringtails aren't using it. Or do they only consume oak mistletoe? Well, we've only seen it in the oak mistletoe. And there's actually a study from Texas in 1956, I think it was, 56 or 57, that found mistletoe anecdotally. They didn't actually list it in their food habits because they were looking at stomach contents, but they list it anecdotally being in scats, but they don't give you any idea of how often it was found or anything like that. But that's the canebrake country, which is full of oaks. So that might be the oak mistletoe too. Michelle O'Brien, we have them at Stone Zoo outside Boston. Really? That's cool. Oh, so Stone Zoo has them. Well, I'll have to do a trip to Boston. I'm trying to see every ringtail that I can. Jazzy, nice to see you too, Jazzy. Kirk, what explains their ringtails? And do you say the white part of the way is more white on the underside? Yeah, so the white part's more on the underside. Well, you have to look at it as disruptive collection. Now, Gene has talked to me about this where he's seen, and I've seen it in my videos too, that they'll whip the tail around when they're unsure about something. Like when I was using the infrared video cameras that were not the black infrared, they would go over and look at them and they'd whip their tails around. I think that's a warning that there must be other ringtails around and that they're saying, hey, there's something here. It's like pronghorn antelope, how they'll lift their tails. And that white is really visible. It's a flashing kind of color. But Gene also was talking about how ringtails will flip their tail over their body when they have to go across open areas. And I think I remembered Michael saying that ringtail, when it was moving across the open area, was flipping its tail up too. So there's a guy named Marty Cordano, I think it was Marty, that reported a ringtail getting almost predated by a great horned owl, where the great horned owl came down. The ringtail had the tail over its body, not flush to its body, but above its body. And when the great horned owl came down, grabbed the tail, and the tail just slid out from between the talons, and the ringtail escaped. So that might be another thing where that white makes it more visible, more of a target, so it gets hit. But ringtails typically stay undercover, so they don't have that issue as often. Laura, good to see you too. Jesse, ringtails, can't wait to see them again. Yep. Curious about your eco tours. Oh yeah, so yes. Oh, Catherine, this is great to hear from you guys too. Stick around after the talk. I want to email you, send you a note. Bree, we have them at Pueblo Zoo in Colorado. You're their trainer. That's awesome. I've actually got on my wall at school a painting that was done by Mala, the ringtail at Coyote Point Museum. And what they did was they would give Mala a little paint palette, and she stuck her paws into it and then would walk over canvases, and you get a ringtail original. You know, so I've got one of those too. So that looks like that's it on questions.
But Dave, I have some questions too.
Yes, please.
So you mentioned that there are no mountain lions or bobcats in the Sutter Buttes. That seems kind of unusual. So why are there no cats in the Sutter Buttes? And would cats be a significant threat to ringtails in other places?
Well, I would say in general, mountain lions, there's a fairly hard no on them. And it's probably because of the cattle operations. So what they have documented are mountain lions traveling through the riparian corridor of Central Valley, hopping through the Sutter Buttes and then going to the Coast Range. So that has been documented. In fact, I do have one photo of a mountain lion that did that. But I've got about 5 or 6 photos of bobcats. So bobcats are a little bit more prevalent, but not in large numbers. And it might be, you don't really have a population of wood rats. There's a very tiny population of wood rats. The only thing there are in terms of rodents are some Peromyscus and Microtus. And I think there's just not enough food resources for them. I don't think there's just enough cottontails to support any sort of decent sized population of bobcats. I think the bobs have to move substantially around that area, and even at 46,000 acres, it's just not enough habitat, I suspect, for bobs. So I don't think bobs stay there very long. Yeah, which leaves it for the smaller mesocarnivores. Yeah.
In our Tuleyome property up in Lake County, we've got a bunch of game cameras, and we've picked up a lot of mountain lions and bobcats, but never a ringtail.
Yeah, I wonder if they're getting eaten. And the fires have been pretty detrimental to them too. You know, the fires have been problematic with them. I saw I had a question that came up. Do you lead programs with the Middle Mountain Foundation? Yeah, and it's actually called Middle Mountain Interpretive Hikes now. So if you go to that website, Middle Mountain Interpretive Hikes, they'll list the hikes. They're hoping to resume the spring program. I usually in the springtime do a opportunity for people to join me on my bat research out there. And then sometimes in the spring, I'll do a ringtail outing. But most often it's in the fall, and I do it in conjunction with Gene Trapp's wildlife hike, where he does a wildlife hike and then you swing by. I set it up so I do ringtail work that weekend, and you swing by and visit with us with the ringtails. And then Brian Hausbach and I will lead a biology and geology hike in the Sutter Buttes as well. But there's a whole bunch of them, like photography, Native American, ethnobotany hikes, hikes that are hardcore hikes up the ridges and so on too. One of the ringtail trainers at the Living Desert in Palm Springs here. Oh, good, Jamie, excellent. Yeah, the Living Desert has some beautiful ringtails. I've got some great pictures of, I think you guys had like 4 babies one year, and there's 4 of you holding the babies, and they are cuter than heck. Yeah. So another question, Bill.
I think that was it.
Okay.
How many babies do they have at a time? Is it typical to have 1, 4, more?
Normally it's a 2 to 4 range, with 2 to 3 babies being the number that they had. Yeah, I have a humorous story to tell too. I was down at Sonora Desert Museum, and they have in their nocturnal exhibit, they have ringtails. So I was getting ready to go over there, but the geology exhibit wasn't very busy. So I went through the geology exhibit. As I'm walking through the geology exhibit, there's a docent there, and I'm the only person there. And I come to this screeching stop as I'm walking by it, because there's a baby ringtail in the exhibit. And I'm like, there's a baby ringtail. There's another baby ringtail, and there's a female. And I turned to the docent. I said, you have ringtails in your geology exhibit. And she starts laughing and says, those are wild ringtails. They actually broke into the exhibit and made their den between 2 of the exhibits. So because the ringtails were so, they didn't care if people watched them. They just went ahead and replaced it with a glass so people could actually see them. So I ended up spending an hour just watching these babies wrestling with each other, and then napping, and then wrestling and sucking on their paws. And they were so cute because they still didn't have their ear cartilage, so they were just flopped down. They were amazing. Yeah. So that was fun.
Okay, well, thank you so much. Really appreciate your talk. If you want to stick around and talk to some of the friends you have on here, I'll leave the meeting going.
Yeah, please do. And then I could stop. Do you want me to stop the recording? Actually, I can.
Yeah, I'll go ahead and stop the recording.
Okay, and then I could. I'll stay on, and if people wanted to